So, very warm welcome to the Young Investigator Day of the CRC 1182. It's very nice to see so many of you. It's a wonderful occasion in these times to be together, at least virtual, together with you. 1 year ago, December 2019, we all were very excited because we just were anticipating a new funding period. The CRC was evaluated by an external board of reviewers. The DFG Senate decided to follow the recommendations. So this place here, the CRC 1102, was an excellent place for doing research in the coming 4 years. In December 2019, many of you were excited because they have applied for new jobs, graduate students, postdocs, and so they were looking forward to move from their country, from their city, from their environment into a new adventurous environment in the north of Germany, to a place called Kiel, in a research center called Origin and Function of Meta-organisms. And the world around us was also very exciting because the research we are doing in host microbe interactions is an uprising, is a new field. Big papers are coming out, money is there, colleagues around the world start to jump on the train, and exciting times ahead of us. And there was nothing which could change that. Many of you decided to move to Kiel in this northern city, and you got acquainted to your PI, you got acquainted to the many model organisms which we are working to understand the functioning of a meta-organism. And you arrived here and you found Kiel, well, not so bad. It's not San Francisco, it's not Berkeley, it's not Barcelona, but it's a good living there. It's okay, good for outdoor activities, and the research is wonderful. End of February 2020, beginning of March 2020. All plans were disrupted by the COVID pandemic. The virus was never on our agenda. It was never on our second funding period perspective. But the virus started to drive our agenda. All of a sudden, we had to deal with immediate halts of face-to-face meetings. We had to work remotely. We are staring from 8 in the morning till 9 in the evening in the computer screen. We are accepting that most or all of our meetings were postponed or indefinitely suspended. We are holding virtual conferences, listening to Zoom seminars, meeting our CRC teams online. I think we all were surprised how much we miss the personal interactions, how much we need each other, how much we take for granted in terms of social interactions and opportunities. Our research focused on understanding the interactions of meta-organisms in all its facets is a deeply social enterprise. We need the input from our team members. This is not only useful, it's not only important, it is necessary. And as members of the CRC, we are deeply connected to all others, the juniors, the seniors, and those connections sustain a collaborative research center. all of a sudden it seemed that we had lost at least part of what sustains us as a collaborative research center. Today, we are celebrating the Young Investigator Day 2020. This also was not on our agenda. The awards in the last few years were presented in the shadow of the big Möbius Fellowship ceremony. And so at the end of the Möbius ceremony, the young investigators got their prizes. Our fellow this year is Rob Knight from San Diego, and we decided to postpone this ceremony to next year. But we also decided not to postpone the Young Investigator Awards. We wanted to have a day exclusively and deliberately focused on the young investigators, on you, for many reasons. And I want to thank you for being with us and for working so efficiently and even so closely together in these confusing times. I want to thank you for what you have achieved so far in this first year of the second funding period. I want to thank you for keeping going and participating in our, of course, online activities, in particular in the meta-organism seminar. I was so much surprised when I looked into the first meta-organism seminar after lockdown, and I saw more than 30 young investigators participating, presenting brilliant ideas, new observations, asking good and critical questions. Thank you. The Young Investigator Day also should indicate that we, the senior members of the CRC, the steering committee, is taking care of you, and believe me, we feel responsible for you. We know how difficult it is in this time. Just having moved from abroad to Kiel, knowing nobody in this city, starting a new project, getting locked down. But remember, we are teams, and we live in teams. As CRC, we have not only introduced an online format for the meta-organism seminar and today's Young Investigator Day to highlight and appreciate the work of the junior generation. To cheer you up before Christmas in these not so Christmas-like times, Annika and Cleo have organized an online coffee shop and an escape room, whatever that means, in the coming weeks before Christmas, and I strongly invite you to join that. The CRC Steering Committee has announced 2 Creativity Awards, €5,000 each. Deadline next week. Please submit your ideas. It does not have to be a shining, glossy, complete, ready-to-go product. Submit your idea, your concept, anything which opens up new thinking on the meta-organism is happily welcome to be received, and then you may have a good chance to get €5,000 to continue your creative work. The young investigators in the CRC also have elected a representative in the steering committee. It's now Malte Rühlemann. Malte is a wonderful person, and Malte is waiting to get your emails and to get in contact with you if there is a problem which you think a team member of the same generation of a similar career stage may help you. And Malte is currently with us here in the Zoom meeting, but he sits in Leuven in Belgium, and but he is really waiting if somebody has a need so that Malte can somehow help. And we have Ute Jöli, our wonderful coach and mentor in Heidelberg sitting, and also Ute is here today. I can tell you that Ute's thoughts are every single day with us here in the CRC. And I can tell you, if you have particularly some more intimate, more personal worries, please address Ute. I know her for several years now, and she works in a wonderful, in a magnificent, discreet way, and tries to help particularly the junior generation. The coronavirus has caused the loss of some of human bones. We of course value the technology, the technological progress, what we can do to get very deep in microorganism details. But what defines us as human beings is closeness and community. And I very much hope that we will come back there again soon. With that few words from my side, I'd like to go on in the agenda. And today's program has 4 talks. 2 brilliant junior investigators from our center were selected by the steering committee to be awardees of the 2020 CRC Young Investigator Prize, 15,000 euro each. And this is Lucía Galán and Malte Rühlemann. They will present now their research, their ideas. I have no idea what they really present. I'm very much looking forward to that. There will be possibility to ask questions. It's a scientific talk, and so they got a big sum of money, and now let's see how good they really are and what clever ideas they will present us and what they talk about. And after that, we decided to round that day up, that we invite last year's awardees for a little shorter presentation to tell us what happened since last year when they got the prize, and that's Shawnee and Jinru. And so they will then, after the awardees, also give a presentation. It's really awful as human beings, because a ceremony without flowers, a ceremony without getting a kind of a sculpture or a prize and a certificate, and a ceremony where we cannot go out for dinner with the people is awful. But that's the way it is at the moment. So my last hope with Hinrich and the steering committee that we at least can go out with the awardees for dinner, but that is also not possible. So Cleo has organized a bunch of flower to the awardees of this year, and they also will get their little wonderful precious plastic sculpture, which reminds them for the rest of their career that they were awarded the CRC Young Investigator Award 2020. But we are all here kind of virtual. Having said that, I don't see Malte at the moment, but I would like that Malte gets...
I'm here.
Malte, are you ready to upload your talk? Oh, I'm sorry, Lucía comes first. I'm sorry, ladies first. I have nobody in my room who helps me, and so I am helpless. Lucía.
I'm here, I'm ready.
That's wonderful. So first, in the name of the steering committee of the CRC, I want to congratulate you. I give you the bunch of flowers. Did it arrive already?
Not yet. We think may arrive during my talk, actually, but I have here the prize.
Okay, maybe you Lucía, show it in the camera so that everybody can see it. So the flowers are in the car with the driver, and the prize is in Lucía's hands. And so we got a number of applications, and the steering committee decided to give Lucía and Malte this year's prize. And so very briefly to introduce Lucía Pita Galán. She comes from Spain. She did her PhD at the University of Barcelona. She did a postdoc in Würzburg. You can guess with whom. And then she moved to Kiel. She got awarded a Alexander von Humboldt postdoc fellowship for 2 years. Very nice and very prestigious. And she continued as a postdoc with Ute Henschel here at the GeoMar. And since April 18, she's a junior group leader at the Marine Symbiosis Unit in the GeoMar. Lucía, you're a wonderful member of this CRC. You're manifold connected, writing lots of multi-author papers. I guess we just finished one the last few days. And it's wonderful. I congratulate you. And the talk will be now, do microbes hijack ancient signaling networks involved in animal tissue homeostasis? And Lucía, the floor is yours for your presentation.
Yeah, thank you very much. Before sharing the screen that I can see you all, thank you for Thomas for your words and Cleo for organizing also the flowers that are on the way. And thank you all also for coming today. I know you are in seminars and meetings in Zoom all the time, so I really appreciate that you join today. So I will now share my screen, and I hope it will work fine.
You have to go to full screen.
Yeah, it should be, it's now okay?
Yes.
Okay, perfect. So now I don't see you. If you have questions, maybe you can ask them at the end, but if something is not clear or the connection is not good, please just let me know. Okay, so thank you again for this Young Investigator Award that is supporting one research idea that comes into this project entitled, Do Microbes Hijack Ancient Signaling Networks Involved in Animal Tissue Homeostasis? And I highlighted here 3 keywords. The first one is microbes, and I think I don't need to convince this audience why looking into microbes and microbial communities is important when we are interested in animal function. The second word is ancient. As you all know by now, I work with marine sponges among the first animal groups that appear on Earth. And they can help us understand, conserve signaling networks that are important for animal health. And in particular, to homeostasis, this is this third keyword, which is the capacity of an organism to maintain constant internal conditions via feedback loops that guarantee that the cells, the tissue, the organs can work properly. And for looking into homeostasis, what I suggest is to a context in which homeostasis is disrupted and needs to be reestablished again, and is that of wound healing. So you may think about healing when there is like a big trauma, and regeneration would be actually positive, but even in small wounds that we all had as a kids, as an adults, healing is also crucial. An open wound is an open door to opportunistic microbes, to pathogens that may cause systematic infections. Also, there are some human diseases, such as diabetes, that interfere with this healing process, and micro wounds may even become ulcers and cause actually very negative problems. There is another link between wound healing and health that is, however, less evident, and is that of cancer. So in 1986, Deborah had this bold idea of defining tumors as wounds that do not heal. What he meant is that there are certain parallelisms between the process involved in cancer and the process involved in wound healing. And this is an idea that has been revisited in recent years. For example, there is sustained proliferation during cancer that is also found in healing, or evading growth suppressors, so keeping growing, or, for example, inflammation. These are processes that are necessary in wound healing, but also occur during cancer. The big difference is that in wound healing, they are tightly regulated. So they are put in place when they are needed, but then conditions go back to the baseline. Whereas in cancer, these processes are completely dysregulated and out of control, and that's why they are causing trouble to the animal. Importantly, many of these processes are actually fundamental of what makes an animal, and we can date back the origin of the genes that control those processes to the origin of multicellularity. These are new genes, new gene families, new gene connections that were necessary to control cell differentiation or cell communication, at the end to have a coordinated cellular behavior. But modern animals, they also have genes that date back to our unicellular ancestor. And these are genes that were more related to metabolism or proliferation. And what Trigos and collaborators and others suggest is that in a healthy animal, the multicellular programming dominates to have this coordinated behavior. Whereas, for example, in cancer, it's more this unicellular programming, as I say, proliferation or more selfish behavior that dominates. And what this implies at the end is that looking at the origin of a gene and also the origin of the gene-gene connections, we can actually find interesting candidates or targets for cancer therapy and to understand homeostasis. For this reason, I think sponges are an interesting group to look into the evolutionary path of the genes involved in healing and in homeostasis. As I mentioned before, these are among the earliest animal groups that emerged in Earth. They also have interesting features. One is that they present high healing capacity, although comparative to other model organisms, they have not been so widely investigated in terms of regeneration, but they do regenerate. And so far, there are no reports of tumor in sponges, which is also quite interesting. So we started to look into the sponge response to wounding in a study that is completely different context, because it was an ecological perspective. And this was the work of my former PhD student, Yu Chen Wu, who was interested in how sponges respond to grazing. She investigated the Mediterranean sponge Aplycina aerophoba that you can see here. Is one of our favorite sponge pets, if you want. And this little guy, this sea slug called Tylodina perversa, has specialized in living and feeding on the sponge. So she wondered how is the sponge sensing this grazing and responding to the damage. I have added here a video so that you can see if it works. Well, I wanted to show you how the experiment looked like. So this is a specimen of the sponge. This is the animal, and we let the animal feed on the sponge for 1 day. And at the end, as you can observe, the sponges could be actually very damaged. We can easily see where the animal was feeding. Even here, we have another smaller scar. In this case, the damage is quite impressive, because the exhalant pore of the sponge, what we call osculum, has been destroyed. So this sponge is not pumping, means it's not also feeding, or it's not a normal functioning. But 3 days after the stop of the treatment, so the animal, the grazer was not feeding anymore, we can already observe that the sponge has built a new osculum and is pumping again. So there is already some regeneration, although we can also still see the scar as well. So then was looking at the cellular and chemical responses involved in this process, but we also were interested in host gene expression, and we ran RNA-seq study that I will present now some of the results, and this is still unpublished. So you see here we have actually 2 wounding treatments. G stands for grazing, so this little grazer was feeding on the sponge. M stands for mechanical damage. So here we damaged the sponge with a scalpel. We were trying to imitate the damage of grazing, but in the absence of the predator. We have 3 different time points, 3 hours, 1 day, and 6 days after the stop of the treatment. And here you have the number of differential expressed genes. That means of gene expression in grazing compared to control, which were intact sponges, and mechanical damage compared to control. The first result is that we find activation of gene expression. So most of the genes were upregulated, meaning they have a higher gene expression level than in control. The second striking observation is that already 6 days after the stop of the treatment, the gene expression levels in grazing and mechanical damage, they are similar to those in control. And also most of the signaling of this response happened in this 3-hour time point. The genes involved in this signaling I have summarized in a figure, and I will walk you through it. So this is a cell with the membrane and the nucleus. We identify many differential expressed genes involved in calcium signaling and calcium dependent. We also have a coagulation-like cascade, if we can call it like that. It's like mediated by transglutaminases and genes involved in extracellular matrix rearrangement. We also find multiple receptors like the G protein coupled receptors involved in sensing the environment. The Wnt signaling pathway that is highly conserved, kinase cascade, and different families of transcription factors were also elevated. I did compare the coding sequences of these differential expressed genes to the reference proteome of Amphimedon queenslandica. This is a species from the Great Barrier Reef. It's not closely related to the sponge species we work with, but it's the reference species for all the genomic studies for sponges so far. Its genome was published in 2010, and it remains the most complete genome for a sponge, and is present in some of the typical public databases. So what you see here is a protein-protein network generated in the program string. Each node represents a protein, and if 2 nodes are connected, means that they are evidence that one of these proteins is affecting the other, or that the genes coding for these proteins are co-regulated, or they appear also closely in the genome. I mentioned before the Wnt signaling pathway, and there are evidence for sponges, for Amphimedon, that is involved in development, and also more recently in regeneration, also in healing in our study. And this actually agrees with what has been found in other animal groups, particularly those with high regenerative capacities. We have also the kinase cascade that I mentioned before, but what I would like to emphasize is that in this case we have evidence for sponges of this relationship, but for the other cases they are based on what is known in other organisms for proteins that are similar to these ones. So that makes me think that these are quite conserved. And for that, I decided to look, okay, what if instead of comparing to sponge species, I compare to humans? This is a biggest jump, but it's still interesting. So what you can see here is again this protein-protein network. Remember, its node is a protein. And I highlighted here what you can see in bold are genes, proteins, in fact, that are coded by genes that are involved in cancer. So there is like a catalog where you can find all the genes that have been reported to be involved in cancer. It's called COSMIC. And these are well-known genes that have been related at least with cancer process. I also coded by color the origin of these genes. So I mentioned before being ancient may be important for play a role in cancer. And Trigos and collaborator dated which these genes are of unicellular origin or more of the multicellular origin. And more important is not only that this gene is of unicellular origin, but also that plays a regulator role in this unicellular programming. So the unicellular ones you see in green, the ones that date back to the origin of molecularity are presented in yellow. And then you see in pink, this is genes that actually mediate this balance between the unicellular and multicellular programming. And in this case, is the mitogen activated protein kinase 3. And this is a well-known gene to play a role in cancer. So what I want to show you with this is that we have preliminary and strong support of genes that are involved in the response of sponge to wounding and how they have an ancient origin and have been highly conserved. Of course, the question now, and that's why we are now part of the CRC, is what about the microbes? So I have focused so far on the sponge, but what is the role of the microbes? And that's why I decided to apply for the Young Investigator Award and look into this project from the CRC lenses. So this clock represents the time of the planet Earth, and you can see here when the life emerged was microbial, and it remained microbial for most of the time. So when these multicellular organisms first appeared and start evolving, they did it in a microbial world. So I hypothesized that these gene and signaling networks I've been talking about must be able to interpret microbial signals. And also that at least some microbes may have evolved to modulate those ancient genes and networks, maybe for their own good, and sometimes maybe also promoting holobiome homeostasis, metaorganism homeostasis, but also maybe in the context of disease. So the first aim of the project is to look at the microbiome dynamics upon wounding by ways of 16S ribosomal RNA gene amplicon sequencing. So we have already some samples that I think we could look into that and see what is happening to the microbial dynamics over time. And in particular, I would like to look into this Candidatus Synechococcus spongiarium. So I showed to you these pictures before where the scar where the C-slag was feeding appears as bright yellow, whereas the intact sponge, the intact part of the sponge is more brownish. This difference in color is caused by this cyanobacterium, Candidatus Synechococcus spongiarium, that thrives in the outer layer of the sponge. This is how the cyanobacterium looks like in the under the electron microscope. It has these thylakoids that are very characteristic, so we can distinguish already only by morphology. And what you can also see here, and this is very specific or particular of sponges, is that the animal cells occur in close proximity to the microbes. So it's very common to observe the cyanos very close to the animal sponge cells. So I think that's quite remarkable. And because we observe here, this border is very defined at right after the stop of the treatments, but it seems that it gets diffuminated. So I think there is some colonization happening, and I would like to study this interface. I must also mention that in the course of our previous experiment, that was 6 days, we have not observed like the complete recolonization of the scar, meaning that it's not turning yet to this brownish color. So maybe we need to extend also the time of the experiment. A second aim of this project is to investigate the intertwining between these mitogen activate protein kinases, the Wnt signaling cascade, and the microbes. So one idea is to run experiments where we can apply inhibitors or inducers of these signaling pathways. And for that, I was inspired, in fact, with some papers published by researchers of the CRC. And also look at host responses, monitoring healing, and also host gene expression by qPCR. We have now some baseline data, so we can also go for more target genes. And I'm super happy to say that finally we have a phenotype. And this, this is a long, it was a long search for us when we were working with the sponges. They are sitting there, seems that they are doing nothing except for pumping, but their pumping behavior, their respiration is also very plastic and very difficult to use it as a proxy of the health or the stress of the sponge. But here, and I would like to thank Katja Dirkin because she's always emphasizing how a phenotype can help us to move from this detailed molecular data to more function. So here we have a phenotype that we can observe, we can quantify over time. So I'm quite excited that we can better translate this knowledge into function. In usual times, let's say, I would go to Barcelona and run my experiments in a piscina there. These are the facilities at the Institute of Marine Science. They have an open flow-through system, and we built this system from scratch, but now it works very well. You can see here extra lights that we add for these sponge species because of the photosymbionts. This can be also modulated, which may be interesting. And I still hope that by next spring, this could be possible. But I also have a backup plan because we also have nice facilities at Geomar. And thanks to the staff of the Kiel Marine Organism Culture Center, the KIMOC, we also have a system where the is open flow through, and the water from the Baltic gets modified in terms of salinity and temperature to get closer to the Mediterranean conditions. So the system is smaller, maybe a bit more stressful for the sponge, but I'm confident we can still run some interesting studies here if necessary. So I hope I convinced you that sponge wound healing provides a new experimental system to investigate how microbes modulate ancient animal gene networks involved in homeostasis. I'm looking forward to this project. As you know, I'm part of B1. But I think this project also brings or builds new bridges with the C project of the CRC, particularly from work that has been done in the lab of Thomas Bosch, but also in situ by Philip Rosenstiel and Thomas Rödel. They work also with epithelium microbe interaction. So I think we can learn also a lot from them. So finally, I would like to thank Yu-Chen Wu, because she started this project from the ecological perspective and initiated this thinking into new ideas. Ute, of course, for fostering my research and let me grow within the marine symbiosis group. My collaborators, as well as the CRC, not only for the award, but also for fostering these discussions that grow into these new ideas. And finally, I'm happy to take questions, and thank you all for your attention. And I will stop sharing my screen so I can see you all.
Okay, thank you so much, Lucía. I would agree with the decision of the steering committee that the prize is very well in your hands. It's remembering also the creativity award. Yes, it's a wonderful new idea. We have 5 minutes for discussion, and I would just start with 2 comments. One is you started with the hallmarks of cancer, and then wounding and cancer is together. I remind you that there is a wonderful paper by Chanel Arthur, and Chanel actually put the classical Hanahan and Weinberg hallmark of cancer paper into a microbial context. And her paper is entitled, I don't remember precisely, I can send it to you, so the hallmarks of cancer in the era of the microbiome. And what Chanel shows is that every single hallmark of cancer has some microbial connection, and so that keep in mind. And the second comment is, I mean, I see here that Alexander Klimovich is also listening here. I find your approach of going from the unicellular old genes to the more modern younger genes interesting. There is a technique developed in Dieter Tautz's lab, which is called phylostratigraphy. And this technique you may know of. Actually, Dieter and Tomislav Domazet-Lozo measured the age of cancer genes by that phylostratigraphy. You could probably, with some good transcriptome data, use phylostratigraphy to follow your wounding genes. And how much that has to do with human cancer, I would be a bit careful. I know that people try to sell their papers by saying that, but I would be a little careful. But your story per se is wonderful. Okay.
Thank you, Thomas.
Questions from the audience, please. I guess the easiest way would be just speak up. That's the easiest way. Any questions?
Yeah, Lucía.
Hi, Katja.
Thank you very much for this very nice and interesting talk. And I'm also happy with you that you have a phenotype now. But then I missed, in fact, if you have already tested or if you had the chance to look if the microbes, if they enhance or maybe even inhibit wound healing.
Yeah, we have not looked at anything microbial on this. It was really an ecological study. We were more interested in this grazer-response connection, but I think that's the key. And I suggested the cyanos because they are sitting there, but maybe other microbes play a role. So that's why I think I will start with this amplicon sequencing just to see at least what happened, if I can see something, but.
Yeah. And I have another quite detailed question, but when you talk about MAP kinase, which ones are they? I mean, there are many different MAP kinases. Is there a specific MAP kinase pathway involved?
So it's difficult to identify a pathway for sponges because we don't have so much knowledge. So we observe different types of kinases, and actually the kinome of sponges is quite diversified. The kinome of Amphimeron queenslandica is the more diverse in animals. So it's very difficult to tell. As I mentioned, the most similar genes would be this mitogen activated protein kinase, but maybe others also important. So.
More questions from the audience?
Yeah, I have a question. Did you try maybe to test the effect of environmental conditions that might influence how cyanobacteria grow? So, for example, UV, the level of UV or the level of dissolved CO2 in the water. So bubbling your water and seeing if there's any effect.
So here we didn't modify any particular conditions, although when we ran these experiments, the light conditions were a bit lower than what they usually find in situ. We have now this new setup that mimics better what they will find in the sea. But I think that would be also a nice way to modify the role or the activity of these microbes that we can hypothesize are important. But we haven't looked into that yet, yeah. I think there are a lot of things to do. I cannot do everything at once. I need to prioritize, but I'm really excited about this new project.
That's a good wording for the end. Thank you so much, Lucía.
Thank you.
Wish you the very best, and we always are happy to hear from you and about the progress of this really exciting and very nice project. Thank you so much.
Thank you.
Our next and our second awardee is Malte Rühlemann. Malte is in Belgium right now, and again, flowers are on the way, and maybe he has received them, I have no idea. And also he got, oh yes, show them, Malte, please, show the flowers. See, they got real flowers in these unreal times. And Malte also received his little plastic sculpture. To introduce him very briefly, we know Malte. Malte got his PhD here at Kiel University. He works with Andre Franke in the Institute of Clinical and Molecular Biology. He got his PhD with summa cum laude and a fantastic work. He's now a postdoc again with Andre, but currently he's guest scientist in the Rega Institute at the University in Leuven in Belgium. I also would like to congratulate, of course, Malte for his work, for getting this award, but also for his many activities and support of the CRC. He always is aware of what's going on, and then he also volunteered to be elected speaker of the Young Investigators in the steering committee. And as I said in my introductory words, Malte is happy to support you or to listen to you or to do something what he can do to help anybody of you if necessary. Having said that, he has already uploaded his talk, Longitudinal Development and Early Life Impact of the Microbiome isoagglutinine production as modulator of host microbe interactions. Malte, nice that you're here. The floor is yours.
Thank you very much, Thomas, for the introduction, and of course, thank you for the award. I'm very happy that I can present you today the project that I invented for this award, and I think it fits very well our agenda of the CRC, of our project in the CRC, and I will introduce it to you now briefly. In the CRC, I'm in the project that is called Host Microbiome Co-Evolution in the Mammalian Intestine, and this is led by Andrea and John here in the top right corner. And this whole topic of host microbiome co-evolution and how they influence each other is actually one that is very much on top of the current events. And as you can see, for example, from this review that was just published, I think 4 days ago in Science by Taichi Suzuki and Bruce Lay. And here they could show that, or they summarized that, for example, the LCT gene, which is responsible for the lactase persistence, directly influences specific microbial members of the gut microbiome. For example, if people do not have this lactase persistence, there will be lactase available for the bacteria in the gut as an energy source, which will then lead, in this case, to the increased abundance of bifidobacteria. And of course, in people which can digest lactase and split it to glucose and fructose, lactose is not available as an energy source. So in these individuals, bifidobacteria are not as abundant as in the other individuals. And this is actually only one example for these adaptations. So where the microbiome somehow adapts to the host, and they have a whole map of different traits that were somehow adapted to by the microbiome, and also this can lead to some kind of adaptation of the host to the environment as well. So there might be some kind of co-evolution happening. And also we are interested in investigating this a little more, and we are trying to do this with a genome-wide association study. So we are testing different the different variants in the genome of the host. And we did this in 9,000 individuals from Germany and looked at their microbiome. And what we could find is in total 38, oh, it's a little lagging, 38 loci that show to be somehow associated with either community level changes or specific microbial members and their abundance or their presence absence. And we did some additional analysis which might show that there's even a direct influence to especially inflammatory bowel diseases from certain bacteria. But there's one locus which we are very much interested in, and this is on chromosome 9, the locus that harbors the ABO histo-blood group system transferase, glycosyl transferase, for which we could find actually 2 signals in close proximity, one with an OTU that was classified as being member of the genus Bacteroides, and another one which is a member of the Faecalibacterium. So 2 prominent members of the human gut microbiome. And why is this ABO histoblood group transferase so interesting? So just to get everyone here on the same page, so these ABO antigens are present on the surface of red blood cells, and it's a structure of sugar residues, which are first completed by a fucosyl transferase 1 to form this type 2H antigen, to which then, depending on the alleles that are present in the specific individual, additional sugars can be added to form either the A or the B blood group antigen, or if no functioning transferase is available or present in the individual, this leads to the O antigen, where no additional sugars are added. And probably you know that these antigens are directly connected to the blood groups. And people with, for example, the A blood group, they develop antibodies, the so-called isoagglutinines, to the B antigen, because they don't have it as their own. And the other way around, people with B blood group develop A antigens, or antibodies against A antigens. Individuals that have the AB blood group don't develop any of these isoagglutinins, and individuals with the O blood group, so where there is no endogenous antigen present, they develop isoagglutinins to both other blood groups. But it is less widely known that also on secreted proteins, there are these ABO blood groups or histo-blood groups present. So in glycoproteins, there is not the FUT1 fucosyltransferase, which forms the H antigen, but the FUT2 fucosyltransferase. And here it's also possible that this FUT2 fucosyltransferase is not working, and this is actually the case in around 20% of the individuals worldwide. And this then leads to an incomplete H antigen to which no further sugar residues can be added. So also if people are A or B blood group, this will not be added to the secreted glycoproteins. And what we saw in our study is that people that have either A or B antigens on their secretions, they have a higher prevalence of this Bacteroides OTU, which I talked about earlier, in comparison to people which have either no working ABO histo transferase or an in-functioning FUT2. And our findings were also just recently supported by additional functional findings, for example, by in this preprint from Kathleen Arnold from the group of Catherine Lusopon, which could show that Bacteroides fragilis can lead to hemagglutination, so blood clotting, and this is mediated by specific capsular polysaccharides, and this is also influenced on the host genotype for the ABO locus. And in an additional genome-wide association study, which was published, or which is in the pre-print state in medRxiv, they could show in a Finnish cohort that they also find an association of the microbiome with the ABO locus. So this confirms to me that we are here onto something, that there is an effect of the ABO locus on the microbiome, and that they are interacting somehow. But let's take a closer look at the isoagglutinins. Wait, here we go. So these isoagglutinins, they are antibodies, and antibodies usually are not present at birth, but they develop in the first few months of life, and then after a few years, so between 6 and 10 years of age, they usually reach the level of adults. But well, I'm no immunologist, but I know at least that antibodies are part of the adaptive immunity, and they should only be produced after this antigen was first encountered. So the question is, where do they actually come from? And this is now where we developed this project from. And we found something that also develops early in life, which is in fact the microbiome. So early in life, it looks a little different, and then it matures to become the gut microbiome that we usually see in adults. And also from studies in germ-free chicken, actually, there is evidence that germ-free chicken do not develop these isoagglutinins. So it might really have to do something with the microbiome. And this is what we want to investigate. And our hypothesis is that there are specific members of the microbiome that are acquired at some time point, which trigger the initial production of the isoagglutinins in infants early in life. So how do you want to investigate this? we came across an article by where Dr. Julia Pagel from, she's a pediatrician at the UKSH in Lübeck, and also John Baines was a co-author of this study. They sampled children early in life longitudinally, and are analyzing these. And we asked her if they would collaborate with us and share their samples, and luckily they agreed. So what we will do, we will perform shotgun metagenome sequencing on 15 individuals on 5 time points from shortly after the day of birth until roughly 1.5 year of an age. And from the shotgun metagenomes, we will try to assemble good metagenomic assembled genomes in a co-assembly approach. And this longitudinal sampling should really help us to get some good input for binning and bin refinement, for which we developed additional algorithms and databases in-house. So we, in the end, hopefully end up with very high quality metagenome assemblies and also high resolution abundance profiles to investigate how the microbiome develops over time. And of course, we need the other side as well. So we need to measure the isoagglutinines. And usually this is done with some kind of antibody titration using agglutination assays, where serial dilutions of the serum are made. They are then added to a column or plate with red blood cells, where you can then see if they are agglutinating, so very strongly here, where they then stay where they are in this column, or if there's no agglutination, they just sink to the ground, and then there are stages in between. And to get the titer, all these dilutions are added to such a column, and so the dilution where there's no longer any agglutination happening is then usually seen as the titer. Another approach is, of course, ELISA. So here, the antigens can be coated to a reaction plate. We have to do this then with BSA-linked antigens, as we're looking at specific sugar residues. And then we can add the serum of the individuals and can then detect the antibodies in using this enzyme substrate reaction. However, the problem is in children, it is not really easy to acquire blood, and also it's not very ethical, and you will not really get this through a review board. So we had to think of a different approach. And then after doing some reading and thinking, we came to the conclusion that isoagglutinins should also, or are very likely also present in feces. Especially when, if it happens as we expect, the trigger for the production comes from something, from a member of the microbiome of the intestine. So we also asked Professor Mark Ehlers from the UKSH in Lübeck to collaborate with us, because he's an expert in immunology and glycoanalytics. And he agreed to support us in the performance of the ELISAs of, on one hand, total immunoglobulin concentrations, but also on the specific anti-A and anti-B isoagglutinins from the children's feces. So very happy to get these measures from there. So our final analysis plan is like this. So what we expect from these experiments is when we have these 5 time points sampled over 15 individual infants, we will look at the microbiome and we will see different patterns. So we will have microbial taxa that are present maybe very early in life, which will then decline in their abundance very quickly here in the blue line, or a little slower looking at this pink line, or which will maybe also stay part of the microbiome. Or, and we will also see members of the microbiome which will be acquired later across this time point. And these might then rise in their abundance, might be very dynamic in their abundance, or they might be acquired, but then will be gone a short time later again, which is also possible and also likely. But using our sampling approach, we hopefully will get a good picture of how this microbiome develops. And then using our isoagglutinin measures, we might be able to identify the actual trigger for where these isoagglutinins are then started to be produced. Maybe hopefully we find signals connected to specific bacteria, for example, this red one here, also this green one here. And for example, for this green one here, it's possible that these taxa are then more or less eradicated by the immune system because they are then seen as something that is not belonging there and is recognized as something non-self by the immune system. So we hope and we think that this might be a connection and something that can influence the whole process of immune tolerance or the self-non-self discrimination over the whole lifetime. So in summary, what we try to see and the questions we hope to answer are if it is actually possible to detect isoagglutinins from feces. So this is the first question. We really hope so. Otherwise, we would have to think about a different approach how to measure the antibodies. And yeah, and then if this works, we want to identify specific members or hope to identify specific members of the gut microbiome that trigger the initial production of isoagglutinins. And if this is so, we are very, yeah, we would think that there are different taxa or different products of these microbial members relevant for the production of anti-A and anti-B isoagglutinins. And also we might identify specific sets of bacteria that can trigger the initial production. And of course, if this is somehow connected to a discrimination between self and non-self and what the immune system recognizes as something that is part of the meta-organism and should stay part of it, this might really change the trajectory in which microbiomes develop and mature throughout the whole lifetime, already early in life. And with this, I am coming to the end of my talk. I want to thank several people. Come on, yeah. So of course I want to thank my PI, André Franke, for supporting me and also developing my own ideas. But also in general, the whole genetics and bioinformatics group of the IKMB. And very special thanks go to my microbiome subgroup with all the people I'm working there. I'm very happy to be working with these people, and it's really, really nice. In addition, I want to thank John Baines and his group, especially Shawnee Doms and Britt Hermes, who also supported me in the writing and the analysis of the GWAS paper that I mentioned earlier in the talk. I want to thank Professor Mark Ehlers and Dr. Julia Pagel for agreeing to collaborate with us on this project. And of course, I want to thank the CRC for agreeing to fund this idea, because it's probably a pretty high-risk project where it's not really certain what will come out of it. And yeah, I'm really happy that I can at least try to somehow investigate this idea that I had. And in addition, I also want to thank the collaborators for the GWAS paper. To get these 9,000 samples from Germans was only possible in collaboration with the SHIP and the CORA cohorts from Greifswald and Munich. And thank you for your attention. And now I'm really happy to take questions.
Thank you very much, Malte, for an exciting talk. I also want to congratulate the steering committee of the CRC for awarding Malte with that prize. It's clear that this is an exciting novel idea. Variation and phenotypic plasticity is certainly one of the black boxes in our field to understand. There is individual variation, there is microbiome variation, there is host phenotypic plasticity which we do not understand, and so you are now taking one candidate molecule, these isoagglutinines, and trace them for their role in that variability and variation. My only question is, Malte, at the moment, of course, this is a human study, studying with new infants. Still, it will end up as a kind of an association study or a correlation study. Any ideas how to go to causality?
Yeah, so of course, this is a good question. And I think in this whole framework of the CRC, we're somehow now trying to address this whole ABO blood group thing. And we also, in parallel, and this is one of the major projects in the CRC, we are looking at if there are specific, in this case, bacteroides subgroups, which are maybe different and which might be influenced by this. But how we come from causality, from this association to causality is, of course, not easy. Probably we would need some kind of humanized mice in the end that have somehow a similar immune system from which we expect a similar response after maybe monocolonization with specific bacteria. And well, of course, this would be then a whole another story of yeah, trying to get the story a little even more complicated. But yeah, I guess we won't get around to use some kind of model systems. But I think I'd already be happy if I identify some kind of candidates from this approach. And I guess then, and maybe in the next funding phase of the CRC, this might be an approach that we can attack then.
Good. Questions from the audience to Malte? Just speak up if you have a question. I think that's the easiest way. Alex and then Jinru.
Thank you, Thomas. Thank you, Malte. Beautiful hypothesis. Probably I missed something in your talk, but how can you be sure that the antibodies which you detect in the feces are produced by the organism of the infant and don't come from the mother's milk? Because actually the majority of the antibodies, IgA, IgM, in early childhood, they come from the mother. Is there a way to check also the milk as a source of this agglutinating antibody? Do you plan to do so?
Oh, that's actually a very good question, and that's the thing which I haven't thought about yet. So maybe we'd have to control for diet somehow, looking at formula-fed children maybe, but then they would have to be exclusively formula-fed. But also it's possible that yeah, it's actually a very good question. I would have to think about this a little more, and hopefully we can find a way to stratify for that. Maybe we can see already, well, if it's coming from the mother, we would see it already very early, and maybe we see an increase later, or something where we can yeah, interpret that it's maybe really coming then from the infant host. But very good question. I would have to think about this.
Thank you very much. That's why we are together. We on both sides, we are learning during this course. Jinru?
Yeah, thank you. Thank you, Martha, for this very exciting project. I'm just curious, can you validate your discoveries in the future with some systems?
Yeah, thank you. Yeah, as I think that goes into the direction of the question that Thomas already asked. I guess it's probably mice would be then the right system that are somehow being humanized for their immune system. And I guess this would then be the way to go to identify if this is really some specific bacteria, which then can be mono-administered to germ-free animals, which might then provoke the same answer. I guess that would be the way to go. But I'm also not an expert on animal models, so I guess I would have to talk to other people then if I had these plans once I have candidates. Thank you.
Are there more questions to Malte? I don't see this. So then we close the first part of the ceremony.
Thank you.
And we congratulate once more Malte and Lucía. And we thought that it would be nice to invite for a little shorter talks the two awardees from last year to tell us what happened since last year. And the first talk will be given by Shawnee Doms. And so she, just to introduce her very briefly, she got her master in Belgium and then moved now as a PhD student to John Baines, and is a doctoral candidate still, and the project title you see already, Characterization of CYRC4 and its relation to the gut microbiome and circadian rhythms in Drosophila. Shawnee, very nice that you are here, and we hope you have spent the money in a very productive, creative way, and we are eager to listen what happened.
Thank you, Thomas, for the introduction. So yeah, what did I do with this 15,000 euros? So last year I designed this project to characterize CYRC4 and its relation to the gut microbiome and circadian rhythms in Drosophila. And for this, we collaborated with Thomas Röder's lab and Gura, who did the experiments as part of his master thesis. So first, why did I want to do this project is because during the previous funding phase of my main project, we started with mice coming from the hybrid zone between Mus musculus and Mus musculus domesticus. And these two subspecies of Mus musculus, they share a common ancestor about 500,000 years ago. And we see that the hybrids, they have reduced fitness. And this is mostly seen by reduced fertility in males and a higher parasite load. This natural occurring hybrid zone has also long served as a model for evolutionary biologists to study the host fitness. So we, together with Dr. Leslie Turner, developed 8 inbred lines originating from 4 different locations around Munich, which is here, and then we crossed them for 2 generations to make as much variation as possible. And then the plot you see here on the right is the results of our genome-wide study in mice, and shows all the QTLs on the chromosome. And then to prioritize which hits to look at, we used Hannah's geographic screen. So she collected microbiome data from different subspecies and then looked specifically for indicator species for one or the other Mus musculus subspecies. And one that we did have as overlap was amplicon sequence variant number 35, which classifies most closely to Bacteroides acidifaciens or uniformis. And we see that this ASV35 is more abundant in musculus. Then here on the top plot, it's again the abundances from Hannah's geographic screen, and at the bottom are the abundances from ASV35 in my study. And we know that the GG genotype is most common in domesticus and the AA is most common in musculus. So this suggests a genetic basis for the subspecies difference in abundance. And then next we check the expression of the nearest gene, which is CRC5. And as you can see here, according to genotype, this completely mirrors the abundance plot. So what is CRC5? It belongs to a family of highly conserved NAD+ dependent protein deacylases. It can deacylate carbomoyl phosphate synthetase, which leads to the ammonia detoxification. It also controls the glutamine production, and then that's ammonia production. And apart from that, it also is involved in many important pathways, such as the amino acid degradation, TCA cycle, glycolysis, fatty acid metabolism, and so on. it is predominantly situated in the mitochondria, but it can also locate to the cytoplasm. So we can say that it plays an essential role in metabolic homeostasis by acting as a metabolic sensor under the conditions of fasting, high protein intake, or caloric restriction. And it does this in a circadian NAD+ way. Also, the sirtuins are also a very hot topic today, because you can do a sirtuin diet, which apparently includes eating chocolate and drinking red wine, and then you can lose 100 pounds like Adele. Then we go to the Bacteroides acidifaciens. So what we know about this one is that it displays cyclic diurnal oscillation depending on feeding time. You can see this here in this graph. The red line are mice fed during the light, and the blue line are mice fed during the dark. And then the phylum Bacteroidetes is known to be linked with the host metabolism, and it increases on the conditions of caloric restriction and decreases with the excess of energy intake. Then Bacteroidetes also show no cycling in mice that don't have a functional clock, which is this red one. And the Bacteroidetes also increase with greater nitrogen supply. So can the host attenuate nitrogen limitation to upregulate preferred taxa? So this is one of our hypotheses. So the possible interaction between Bacteroides acidifaciens and sirtuin 5 could go through the nitrogen exchange. Here I show a model of cooperative host microbial diurnality. So we have the light, which entrains the host master clock, and then this rhythmicity will tell the host when it's time to eat or rest. And the feeding times will also control the activity of the microbiome, which then gives a feedback to the host by metabolic control. So our goal is to examine sirtuin 5 expression and bacterial abundances in a circadian context. So we can split this in two questions. First, is sirtuin 5 expressed in a cyclic circadian pattern? And then second, is the bacterial load correlated with the sirtuin 5 expression? So for this, we collaborated with Thomas Röder, and we chose Drosophila as a model organism. Drosophila only have one mitochondrial sirtuin, or one sirtuin that has a predicted mitochondrial targeting sequence. So it might be the ancestral sirtuin of sirtuin 5. And then again, thanks to Gura, who conducted the experiments in Thomas Röder's lab. What do we know about the sirtuin 4 in Drosophila is that first, sirtuin 4 seems to have an effect on the lifespan. When they knock out sirtuin 4, the flies seem to have a shorter lifespan, while if the sirtuin 4 was overexpressed, they had a longer lifespan. And here this graph shows the time in days and the percentage of survivors. Then they looked at the effect of starvation, activity, and fertility. So here in plot A and B, they show the effect of starvation with on the x-axis the time in hours and on the y-axis the percentage of flies alive. Then the knockout flies, so the red line here, are more sensitive to starvation compared to the wild type, while the overexpressed are more resistant to starvation. In plot C, they see that sirtuin 4 levels increase upon overnight fasting in fed bodies. And here they also see that the sirtuin knockout flies show less spontaneous activity and are also less fertile on high caloric or low calorie diet. So we had a workflow that we start first with the wild type flies, and we raise them or germ-free, which turn into microbially depleted, or we raise them conventionally by colonizing them with these bacterial strains. And then we will measure the expression of period, timeless, and SIRT24. We use period and timeless because they are genes that are known to cycle, so they are positive control. So here you see the group of conventionally raised flies in blue, and the microbially depleted flies are in yellow. So Kura collected flies every 3 hours. And in these plots, you can see that the expression changes over time. And we tested with GTK cycle the cycling, and all of the genes cycle except timeless in a microbial depleted condition, which is here the yellow. Then we look at the average log fold expression per replicate, and we see that period, timeless, and SIRT24 all are significantly higher expressed in the microbial depleted situation. So we can conclude, yes, SIRT24 displays circadian cycling pattern, and it also has a higher expression level in the microbial depleted flies. Then our next part is we wanted to see if the bacterial load is correlated with the SIRT24 expression. So we have 4 different conditions. We have SIRT24 knockout flies, overexpression flies, knockdown, and again, wild type as control. And we have them in the conventionally raised for 8 time points. So here in this graph, we plotted the relative bacterial loads of each of the conditions. And the peach graph here is the knockout, and you can clearly see the cycling pattern. The knockdown flies which is the red one here, show a similar pattern to the black, which is the wild type. And the overexpression, they have a very low bacterial abundance. Also, when we tested this with GTK cycle, we saw that the cycling was everywhere significant. Then if we look a bit closer at these curves, together with this bar plot here, that shows the period in blue and phase in yellow. So the period is the time between 2 peaks, and the phase is the time it needs to reach a peak. So the knockout, the peach line, already reaches its peak at time point 0 and has a period of 9 hours. While the wild type here, the black line, reaches its peak at 4.5 hours and also has a period of 9 hours. So it looks a bit like there's a jet lag between the wild type and the knockout. Then the red line, the knockdown, has its peak already at time point 3 and only has 6 hours between 2 peaks. And then for the overexpression, we need to zoom in a bit more because it has such a low load. So you can see here that it reaches its peak already at, no, not already, only at 6.5 hours, and has a period of 18 hours, which is twice as long as a wild type. Then here we show the correlation of the average bacterial loads in each of the conditions compared to the SIRT24 expression. So it's only suggestive, but it shows a pretty good reverse correlation between the expression and the bacterial load. So is there a compensation or less regulation of the microbiome? It's one of the next questions. So we also had planned to compare the flies between conventional and germ-free, but as you can see here, these should be germ-free and still manage to get a very high bacterial load. So this is odd because we handled all the lines at the same time. So maybe some genotypes or conditions were more susceptible to colonization than others. Then to summarize a bit what we know now is that so SIRT24 does show a circadian cyclic pattern, and more expression of SIRT24 is correlated with a lower bacterial load. And Wood et al. Said that SIRT24 expression increases during fasting. He also found that they had the knockouts have a shorter lifespan, are more sensitive to starvation, less spontaneous activity, impaired fertility, and we found higher bacterial load. So I have tried to combine what we know about SIRT24. So we know that the light entrains the host clock, and this will control if they take in food or not. So in case of fasting, then there will be no food. So no food will also have low bacterial load, which we know now is correlated with high SIRT24 expression. And that would lead to SIRT4 succinylating proteins for ketogenesis. And because of the increased amino acid catabolism, there is increased ammonia. So SIRT4 also would need to activate CPS1 to start a urea cycle to get rid of the ammonia. And then if there is food, then we would have a high bacterial load with a low SIRT4 expression, which means that the SIRT4 is not there to suppress the cellular respiration. So there will be a high cellular respiration with high glycolysis and high TCA cycle, and they can produce a lot of ATP. So this also shows this cooperative model that we had in the start, that it's supported with what we find now. What do we still need to do with this money is that we still need to do 16S RNA gene sequencing from the conventionally raised flies, and we also still need to have all the SIRT4 gene expression from the different conditions. And with this, I want to thank my group and Thomas Röder's group for the nice collaboration, and thank you for your attention.
Thank you very much, Shoni. Very nice. Also a very hot topic, circadian rhythms and microbiome. It's known that changes in the circadian rhythm, shift workers have chronic diseases, and so the microbiome seems to be also a major player. I'm not sure if just to start with the questions, Shoni, what you clearly could show that there is a correlation between SIRT4 and bacterial load, and if you knock out the gene, then you have a major disturbance. My question was probably what you mentioned at the very end of your talk, the other way around. Do microbes affect the Drosophila clock, the host clock? You tried to do germ-free flies, but it didn't work. There are two sides of the coin, and so the host has certainly influence on the microbiome, but the microbiome may potentially also drive the host clock.
Yeah, so that's one hypothesis, that it could be through the nitrogen exchange. So because the SIRT4 will have a lot of excess nitrogen that then the microbiome can use, or the microbiome has a lot of excess nitrogen that then will be through the host. So we think this is one of the possibilities. How we look for this, and we also wanted to do more experiments where we had fasting conditions in the flies, but we didn't manage this yet. So I think in these ones we could also have more of an insight in how the fasting and the microbiome goes together.
Thank you very much. Questions from the audience to Shoni? Henrik.
I'm particular. First of all, thanks, Shoni, for your very nice overview of the work. I'm still intrigued by the results for the knockouts of the overexpression strains, because I mean, you could assume that if you manipulate the gene regulatory network determining sort of circadian rhythms, that you simply get no rhythm at all. But it appears that your the cycling you get is still very synchronized. Could you perhaps explain that in a little more detail? Is that really true, or do you actually, in the knockouts, for example, do you actually get a lot of variation? And if not, is there any way, any ideas how to explain that it is really a clear-cut period apparently in the knockouts?
So the SIRT4, of course, is not the only one influencing the clock. So there's also the other more important clock genes. So we think actually that the SIRT4 might be the more peripheral clock, which would feed back the feeding time, while the other ones like period and timeless are influenced by the light. Then how, if there's a lot of variation, so within the replicates, we already saw a lot of variation in the cycling. which was also, I think, visible on the plots because they had very large outliers. Why is this? I don't know.
Okay, but that you find a lot of variation for me actually makes more sense, and I actually missed that a little bit, perhaps in the summary bar graphs. So it could be worth perhaps to add error bars or look at variation per se, because I think that would perhaps make more sense.
Yeah, they were in the box plots, but yeah, it definitely, because also one point there is actually 7 flies together to have enough material, and then between those replicates we still have quite some variation.
Thank you.
Thank you. More questions? Shawnee? I do not see. So then we would move to the last talk of this Young Investigator Day afternoon. Again, Jinro He. Jinro was awarded last year the Young Investigator Award, and will upload his presentation now. Jinro got his education in China, in Xiamen University, and then moved, got his master there. And then he moved to, he even shows a picture from last year, with the flowers, with the real flowers. And then he moved to my lab and did his thesis there. He graduated, if I remember correctly, in October, and he's now a postdoc in my lab. Jinro, it's your time.
Thank you, Thomas. Thank you, everyone, for still staying with me. Today, I would like to give an update on the yard money that you guys offered me last year, especially with the update report on resolving these hydromicrobe interactions at single cell resolution using a pilot single cell and seq technology to serve our goal with this phenotype. And I'm from Thomas Bosch Lab, and we are working on the C1 project of the CRC. So first of all, I would like to...
Don't go over it. We want to see. Show the picture. So this was last year. Wasn't it nice? How beautiful. Beautiful flowers, certificates. And I guess you also got your plastic remember sculpture. Very nice. Okay, then just, yeah, thank you.
Yeah, thank you, Thomas. First of all, I would like to thank the CRC committee and also the CRC family for offering me this money so that we can do some kind of adventurous projects. And just briefly to bring everybody, on board. I'm working with Hydra. Hydra is a fruitful model organism for developmental biology for over 280 years. And we knew a lot about its basic body plan, its cellular composition, and the gene regulation regarding the development in the last decades. However, all of these were done with the ignorance of the importance of microbes. In the age of meta-organism concept, now Hydra has been developed as a model meta-organism for developmental biology. And one specific question to me 4 years ago was to address this specific question: does Hydra microbiota affect host development? And not surprisingly, since I've shown this picture already multiple times, we discovered this phenotype where, in the absence of microbes, the germ-free Hydra stopped budding in a long-term cultivation status. And also not surprising, we followed these non-binding animals, and we found that after only 2 days of starvation, the same individuals could give rise to multiple bars along almost the entire body column following this treatment. And this phenotype, together with the non-binding phenotype, showed us the basic concept that development and patterning is disturbed in the absence of microbes. We would like to address this question with more fundamental molecular mechanisms. And the first thing we did was to pursue a more traditional thinking that is using the bulk mRNA-seq experiment. We collected all the animals we see here in this graph, and we performed a bulk mRNA-seq experiment. And what we saw was actually extraordinary. First, on the x-axis of this NMDS plot, we could see a clear separation between the animals without microbes and the animals with microbes. And on this left panel, we could see all the germ-free animals were clustered according to the budding status. This gave us a huge hope to look into the deep molecular mechanism. However, we ended up with 210 candidate genes. And those genes, many of them are known to us. Actually, around 60% of them could be annotated. And they span a lot of functions or compositions in terms of molecular function and cellular composition. And when we performed functional annotation enrichment analysis, we could see many signaling pathways. For example, what Lucía could be interested, the Wnt signaling pathway, and also to us more interestingly, this gene transcription regulatory machineries involved in this enrichment analysis. This gives us an opportunity to form up a hypothesis that possibly Hydra microbiota could affect gene regulation, gene expression or gene expression regulation in Hydra, which means Hydra stem cell differentiation process. And this stem cell differentiation process, once it's disturbed, then our later phenotypes, for example, budding and patterning processes could be revealed. With this in mind, we think that we could use this single-cell RNA-seq to actually address how the stem cell differentiation processes are disturbed in the presence and absence of microbes. And we proposed this project with two packages, actually. And the first package was enabled by a former colleague of ours, which now who now goes to the US. And he, Zibert et al. Published last year with a full package of wild-type Hydra single-cell expression atlas, which they could identify dozens of different cell types according to its gene expression patterns. And with this, we overlapped our bulk mRNA-seq expression data with this single-cell data, and we identified many cell type specific genes. And we focused initially on two populations. The first is this ectodermal food specific genes, and the second, the nematoblast genes. Due to the time restriction, I will focus today on the ectodermal food specific genes. In addition to the first formal analysis with these marker genes, we actually found more food specific genes involved in differential expression between the germ-free groups and the animals with microbes. But to get a deeper understanding of what exactly the genes are doing, we performed an in situ experiment with one candidate gene, which is called the rhamnose binding lectin, CSL3. You could see here from the wild-type conventionalized animals, this gene is strictly restricted to the food region. And the same restriction goes to the germ-free animals who still remains budding capability. So you could see here in the food of the germ-free budding animals, this gene expressed in the food and also in the newly formed bud, which is about to release, so a grown small hydra. However, in these non-budding germ-free animals, we saw the gene expressed along the body column. For example, here one dot, and here a developing center in the middle of the body column. This is quite interesting, but we need more evidence, of course. That's why we went following another protocol to stain the animal with this detection approach to detect the peroxidase activity, which is typically identified also in the developing food. So for example, again, in the germ-free conventional wild-type animals, the food region, including the newly emerged food in the almost grown buds, this region is stained. However, in the non-budding germ-free animals, this region again shows an expanded peroxidase activity. We could get an idea now after this overlapping data sets with the single-cell RNA-seq cell atlas. And we think that now what a normal animal could do to develop new bud or new head structure from the body column is now blocked. Instead, it goes in this direction actually to make the body column more foot-like. This was not possible for us to realize without the single-cell RNA-seq data, of course. But still, we need to know how is this transition regulated, or how does all this happen? Therefore, we proposed the second package included in this single-cell RNA-seq experiment. That is, we want to do a de novo single-cell RNA-seq experiment, where we plan to use 10x genomics chromium single-cell expression technology with 6,000 cells per group, and each cell we plan to sequence 35,000 reads. We would like to actually focus on 3 animals. The first, the wild-type animals, which are able to bud normally, and the germ-free animals which are still has the ability to bud, and the germ-free animals which exhibits the non-budding phenotype. And with these 3 animals, we aim to compare first the wild-type and germ-free regularly budding animals to get an idea what would be the germ-free signature in general could be. And second, we want to compare the budding germ-free animals with the non-budding germ-free animals to get an idea how budding phenotype is regulated. Of course, this is not a project that could be finished by myself. I have many collaborators, including Alex from our lab, and also Joanna from Professor Philip Rosenstiel lab, and also Dorian from ZMB. In preparing for this de novo single-cell RNA-seq experiments, despite the corona situation, despite the platform overloading situation, we managed actually to do till now 1 trial already, where we used ACME dissociation method. We fixed the cells upon dissociation so that we can get immediately the expression pattern, not to just wait 2 hours later regularly as what the enzyme digestion approach could go, where we could see, using this approach, we could get clearly separated single cells, and we could extract RNA from these cells, and further, we could get nicely seeding a product from these cells. However, we failed to generate usable seeding a product. This was likely due to the KIT issue, because on the website of this of the 10x genomics protocols, this KIT, the KIT 5' KIT we are using is not tested. The reason we use that is because the whole ZMB project focuses on this KIT because they always have this cell surface protein labeling method. So what we are going to do, we will try next time with this certified kits, the 3' kits, and we will create a double trial where we pursue further with this fixation-based dissociation method, and the second, this enzyme digesting method, where we are almost sure that will work as a positive control. With that, I think I'm ahead of time, but I'm happy to take your questions. Before I end my slides, I think I have plenty of time to thank many people. I thank Professor Thomas Bosch and many members of the lab for the support and discussions. I thank the CRC collaborators, Professor Philip Rosenstein lab, and also the ZMB sequencing facility. We have collaborators also from the mathematical modeling projects, which who will help us to get an insight about how microbes affect this using the metabolites that are detected by the Munich lab, which the collaboration is still going on, and hopefully with this metabolomics method and the single-cell RNA-seq, I could provide more information, hopefully next summer, about this YARD-funded experiments. With that, I thank you for your attention, and I'm ready for your questions.
Thank you very much, Jinru. Optimistic outlook and very nice work. Are there questions from the audience to that? Just speak up if you want to know all the details of single-cell transcriptomics from Jinru.
I would ask a question. This is Philip.
Sure.
Thank you very much. Nice presentation. So have you estimated about how much genes do you need to detect to actually show the different cell populations? I mean, with the 35,000 reads, this is some estimation how many reads you have, but do you have any idea about the, let's say, the rarefication curve of detected genes, and how many genes do you actually need to detect the functional states you want to look at?
Thank you, Philip. Very good question. Actually, we had two lines of approaches to approach your question. The first is that we reanalyze the data set from the Zibert et al. 2019. There, they sequenced 3,000 cells with almost in the same sequencing depth. But however, they used drop-seq. That's why in this trial, and the second approach is that we want to address this question with our own trial experiment. So with this trial, actually, we plan to run a nano run with this. Hopefully, when we get the cDNA out into this 10x chip, we will sequence later on this trial to get an idea using this refraction curve to estimate how many reads do we need and how many cells do we need to cover these cell types. But again, from the first approach, I think the cell counts that we proposed will cover all the cell types that at least they published already.
Thank you.
Thank you.
More questions from the audience? Maybe we close the presentation from Jinru so we can see us better. If that's not the case, I would really like to thank everybody who joined us for this very nice an encouraging afternoon. As I said in the beginning, the virus may have taken away a little bit of our in-person and social interactions. I'm very sure that all that will come back. And
for sure.
Having said that, we wish you a pleasant and a relaxing and a wonderful Christmas season. Stay safe, stay optimistic, use the facilities and the opportunities which we try to give you. If you think of something which you're missing, tell us what you are missing so that we can maybe support you better. And having said that, very nice Christmas season, and very much looking forward to seeing you healthy and in good spirit in January 21. And we will see what happens then. There is no point of making any predictions right now. Stay safe. Thank you so much. It was a very, very nice afternoon.